So today we will tackle about gene therapy. So what is gene therapy? Therapy is an experimental technique that utilizes genes to treat or prevent disease. So ginawa ang gene therapy para ang mga sakit na hindi kayang ma-cure or wala pang nakikitang panggamot, ito yung magsisilbing alternative na pwede nilang gawin through injections. So, gene therapy was invented to replace a mutated gene that causes disease with a healthy ones. So, by this, yung katawan ng taong may sakit ay nabibigyan ng kakayahan na makatulong na labanan yung sakit niya through injections. But, this technique remains dangerous and expensive. So, ibig sabihin nun, yung mga may kakayahan lang makapag-afford ng ganitong klaseng therapy ang may kakayahan na magamot yung kanilang mga sakit. At talagang inaalam pa ng mga experts kung paano talaga siya magiging perfect or suitable para sa pag-cure ng lahat ng uri ng sakit.
AGTC is using gene therapy technology to develop long-lasting therapies for people with severe diseases. Our initial focus is in ophthalmology. Genetic defects in over 200 known genes have been associated with poor visual function. Genes are segments of DNA that direct the cell to produce proteins that perform a vast array of functions in the body. Many diseases have a genetic basis, meaning that mutated genes provide incorrect instructions that cause the cell to produce an abnormal protein, or disable production of a protein completely, either of which can cause disease. In gene therapy, a functional copy of the gene is delivered into a patient's own cells. Normal protein that is produced from a functional gene has the potential to correct the underlying cause of the disease and provide restorative therapeutic benefit. Gene therapy uses engineered viruses to deliver genes into cells. These are called viral vectors. Vectors made from adeno-associated virus, or AAV, are especially well suited for treating retinal diseases. AAV is a small, simple virus that elicits only a weak immune response and has never been shown to cause disease in humans. In gene therapy, the two native AAV genes are removed and replaced with DNA that contains a functional gene with instructions to produce a normal protein. The DNA also contains a promoter, which drives production of the functional protein. The promoter and gene are flanked by signals called inverted terminal repeats that allow the DNA to be packaged inside the protein shell of the AAV vector. AAV vectors are delivered to cells in the retina by routine ophthalmic delivery methods, such as intravitreal or subretinal injection. Once administered, AAV vectors enter the patient's cells and deliver a copy of the functional gene directly into the nucleus. Once inside the nucleus, the functional gene can support the production of the normal protein and correct the underlying cause of the disease. Genetic defects in the eye can cause vision loss or blindness, and gene therapy can provide a long-lasting therapeutic benefit. AGTC is using its technology to develop gene therapies for rare, inherited retinal diseases that currently have no treatments. One of these diseases is achromatopsia, an inherited retinal disease which is present from birth. Achromatopsia results in markedly reduced visual acuity, extreme light sensitivity causing day blindness, and complete loss of color discrimination. The retina is a layer of cells at the back of the eye. In a healthy eye without achromatopsia, special cells in the eye, called cone photoreceptors, convert light into an electrical signal, which is transmitted across the layers of the retina and then to the brain for visual processing. In an eye with achromatopsia, cone photoreceptors in the retina are unable to convert light into an electrical signal. The most common causes are mutations in one of two genes, called CNGB3 and CNGA3. In gene therapy, an AAV vector containing a normal CNGB3 or CNGA3 gene is designed to provide the instructions for normal CNGB3 or CNGA3 protein and restore cone photoreceptor function. By introducing a functional copy of a gene into a patient's cells, gene therapy is able to correct the underlying genetic defect that is causing disease. The functional gene, delivered by an AAV vector, is thought to remain stable for many years, which suggests that gene therapy has the potential for long-lasting clinical benefits after a one-time administration of the vector. AGTC has a significant intellectual property portfolio, expertise in the design of gene therapy products, and expertise in the formulation, manufacture, and physical delivery of gene therapy products. For more information, please visit www.agtc.com.
From the video, it stated that DNA-mutated gene with no protein can cause disease. So, a functional gene can protect it or can correct any abnormal protein. So, doon, sinasabi pa lang na kapag ka nagkaroon ng abnormal protein ng uh, ang isang DNA, it can cause severe diseases or different kind of diseases that may cause harm to a person's health. So, dito, nagkaroon ng formation or sa isang vectors na sinabi kanina or yung AAV vectors, parang inaano nito sa loob or nagkakaroon siya ng shell para dun sa loob nun, mailagay yung pinahang in-inject yung magiging pinahang cure dun sa sakit. Pagkatapos nun, unti-unti nang papasok yun sa human cell, mapupunta sa nucleus, single strand, second single strand, and then sa episome. Then dun na dun na gagawa ng paraan yung cell na yon para i-cure yung abnormal protein ng isang tao. According from the video, the first genetic type of therapy is gene inactivation, meaning turning off the abnormal ones. So, kapag tinurn off siya, parang naliles yung mga abnormal gene sa loob ng katawan ng tao. Then, pangalawa, yung gene replacement, or ito yung sasabi kanina na by injecting or fighting the abnormal proteins. So, replacing faulty with healthy ones. Then, next is gene addition. Dito naman sinasabi na mag-a-add ka ng mag-a-add ng mga healthy genes para sama-sama nilang defeat yung yung abnormal na gene na meron sa katawan ng isang pasyente. So, sinabi rin doon dito na there are two delivery system. The first one is in vivo or modified inside the body. Yun yung iniimbento pa ngayon or inaaral pa ng mga eksperto. So, yung pangalawa naman is ex vivo or modified outside the body. Yun naman yung ginagawa ng mga or yun yung traditional way ng pagka-cure ng sakit sa isang pasyente. Yun yung kapag nakikita natin sa ospital na may mga dextrose, doon dinadaan yung mga gamot para tuloy-tuloy na pumasok sa katawan ng isang tao. So, yun yung ginagawa ng mga doctors or mga experts sa panahon ngayon. So, kapag maraming genes or maraming vectors na ipinasok sa loob ng katawan mo, ibig sabihin, there are different gene therapies depending on the cell targeted. Halimbawa, para lang ma-cure yung sa lungs or sa kidneys, etc. It depends on how or on what cells the vector wants to target. From the book naman, there are two types of gene therapy. It could be germline therapy or somatic gene therapy. So, germline therapy involves the alteration of the genes inside germ cells, sperm or ovum. During reproduction, these gametes fuse to form a fertilized egg called zygote, which divides and passes on the outer gene into all other cells of the body during the development of offspring. This therapy alters the genome of future generations to come. So, example nito ay yung halimbawa sa isang family or inherit na sa isang family na nagkakaroon sila ng brain cancer every individual. So, a certain female, ayaw na niya magkaroon pa ng ganung case or scenario yung mga susunod niya magiging anak or next generation ng kanilang pamilya. So, by this, ginamit niya yung germline therapy para huminto na sa generation niya yung pagkakaroon ng ganong sakit. Then, the other one is somatic gene therapy. So, unlike germline therapy, somatic gene therapy, ginagamit lang siya or nag-cure lang siya para dun sa mismong sa may sakit or sa may pasyente. Nag-cure lang siya ng specific disease like bone marrow or other types of disease, unlike the germline therapy na kinu-cure yung next generation or next descendants. And then, according to my research, sinasabi din na illegal ang germline therapy because there are still a tendency or there's still a risk na yung magiging form or yung form ng bata ay abnormal. And then somatic gene therapy is usually used until this day.
You may be curious, What's the difference between gene therapy, cell therapy, and gene editing? And why are they usually mentioned together? Well, all three are fields of research with similar goals in mind. Treat disease by changing the blueprints to our bodies at a microscopic level. Our genetic makeup. But how do they do it? That's where the differences lie. First, let's talk gene therapy. It's the introduction, removal, or change in genetic material, specifically DNA or its molecule cousin RNA, in the cells of a patient to treat an inherited or developed disease. There are a couple ways gene therapy can be administered. Typically, genetic material, such as a working copy of a gene, is transferred into the cell. Once in the cell, the transferred genetic material changes how proteins are created, so they can do the work for the gene that's not functioning. Proteins are the building blocks of how we function, so they make a big impact on our health. A vector, which is often a virus, delivers the gene into the cell. Don't worry, the viral genes that could cause disease are removed. Then the vectors are directly injected into the patient's body. The new working gene is able to alter the behavior of the protein within the cell, whereas cell therapy uses living and sometimes genetically modified cells to help treat a disease. These transferred cells can be from the patient or someone else. Cell therapy is routinely administered through blood transfusions. Genetically modified cell therapy is when a cell is removed from the body and the gene inside is altered, and then it is put back in the body. This combines components of gene and cell therapy approaches. For more on that, here's Dr. Rain Rouse.
An example of this approach is CAR T-cell therapy. We take an immune system cell, usually a T cell, and we introduce a new gene into the cell carrying a specialized receptor that's been trained to only target molecules that are on the surface of cancer cells. Large numbers of these changed cells are grown in the laboratory and then returned to the patient's body so that they can kill cancer cells.
You also may have heard of CRISPR, or zinc finger nuclease, which are types of gene editing. Gene editing is an up-and-coming approach that works with gene and cell therapies. There are no approved gene editing treatments yet, but many are currently in preclinical and clinical trials phases. The goal of gene editing is to remove or correct faulty elements of the DNA within the gene. This can be done either inside or outside the body, using the patient's own cells or donor cells. Gene therapy, cell therapy, and gene editing are ways to treat diseases at different microscopic levels of the body. For more information about these approaches and how they work, visit asgct.org.
For the last topic is what is the difference between gene therapy, cell therapy, and gene editing. So, in gene therapy, just like what I stated earlier, ito yung parang nire-replace yung isang abnormal genes with new healthy ones. While cell therapy, ito naman yung inserted in a lab, or para mas madali, it specifies lang sa mga blood or sa mga blood cells or veins ng tao. Then gene editing, by the word editing, it means removing or correcting an element or a faulty element inside the patient's body. Ang gene therapy, hindi naman siya limited sa tao lang. Ginagamit din siya sa mga animals like monkeys para dun pa mas makita kung paano pa ka-effective or paano pa magiging mas maganda yung pag-produce or pag-cure sa isang pasyente. Then gene therapy, cell therapy, and gene editing were done to treat disease of a human's body. So, kailangan lang naman na magkaroon pa ng faith sa mga doktor or sa mga experts para magkaroon pa ng chance yung mga may sakit like cancers, different kinds of cancers, na ma-cure pa sila. Hindi nila iisipin na wala na silang pag-asang gumaling. And then for my motivational quote, uh, where there's a will, there's a way. So, kung gusto naman talaga nating makatulong sa tao, gagawa tayo ng paraan para makatulong sa kanila in a way na gagaling sila. For the patients na sa tingin nila wala na silang pag-asang gumaling or na napanghihinaan na sila ng loob, hindi nila kailangang mapanghinaan kasi gumagawa ng paraan ng mga professionals para magkaroon pa ng chance na mabuhay or para mapagpatuloy yung kanilang takbo ng kanilang buhay sa mundong ito. And then, that's all for today. I hope you enjoy. Thank you and God bless.